First up, we have Dr. Oleta Johnson. Dr. Johnson is the William R. and Daniel L. Young Career Development Assistant Professor of Chemistry at the Massachusetts Institute of Technology. Oleta's research group employs chemical biology, biophysics, and biochemistry approaches to study conformationally dynamic proteins that aggregate in disease. Oleta?
All right. Hi, it's really exciting to be here. Can we get rid of the title? And anyways, can chemistry be used to direct cellular traffic is what the title says. And before I answer, the answer to that question, we hope in my lab, is yes. But before I tell you a bit about what that means and how we want to approach this, I actually have another question that I want to pose. Oh, thank you. Which is, what do these 5 people have in common? And so, at first, the answer might be, I don't know, they're pretty different. And then you might think more about it and say, well, these people are all pretty notable, they're all pretty influential in their respective fields. But as it pertains to my research program, they all share another unfortunate commonality. So all 5 of these people were diagnosed with a devastating class of disorders known as neurodegenerative diseases that ultimately robbed them of their cognitive and physical faculties that they used to become so influential. And although all of these diseases are quite different, they do share one feature, which is the appearance of protein aggregates, as you can see by the dark spots on these slides underneath the disease. And so what that means at the molecular level is that proteins that are normally pretty flexible and use that flexibility to carry out their normal function, they end up trapped in a conformation that is aggregation prone. And so they clump together and form these aggregates that cause these sort of protein traffic jams. And these aggregates become very detrimental to other processes in the cell. So whether they're correlative or causative of disease, it's important to develop systems that prevent them from forming in the first place. Unsurprisingly, the cell has already worked on this. And so, in part, with my favorite protein, DNAJB6, which is a molecular chaperone protein that carries out its role by 2 main functions. First, it detects those aggregation-prone conformations very early on. And second, it prevents them from aggregating in the first place. But as we see, that doesn't always work out for a number of reasons that I'm happy to talk about later. But this inspires the research in my lab, and we have 3 goals toward that. First, we want to develop new chemical tools to detect changes in protein conformation. Second, we want to learn a little bit more about the mechanisms that DNAJB6 uses to prevent these cellular protein traffic jams. And third, we want to discover chemical directors of protein traffic that can either potentiate DNAJB6 activity or compensate for it when it's not working properly. So I want to spend the rest of my time talking a little bit about work that we're doing to develop new chemical tools, but they also kind of influence learning about how DNAJB6 prevents cellular traffic jams, because DNAJB6 forms these oligomers, they self-associate, and they exchange, so it's hard to isolate any one population, and we don't totally understand how that correlates to its function. So my second-year student, Cordiana, had this idea of what if we had a chemical probe that we could mix in with a protein, and it would covalently label the protein to turn on a light fluorescent signal. And then when another protein, like an oligomer, came next to it, it would cross-link to kick off that chemical probe and cause a decrease in the fluorescent signal. Thus, if we look at this over time, we would be able to get a fluorescent fingerprint that tells us something about how protein conformation is changing over time. Now, this is the part where all the first, group 1 fellows are going to hate me, because I'm not going to show you what that chemical probe is today. But I do hope to have a story about this submitted at the end of this year, and so be on the lookout for that. But what I can tell you is that this molecule that we're calling CC0, at least right now, Cordiana tried this in really mild conditions to see if we could see protein cross-linking and to see if we could see this fluorescent signal. We see something that makes it seem like something is happening, but we need more time points, and we also weren't really satisfied with the dynamic range, so we wanted there to be a bigger delta between time 0 and our maximum signal. So we looked on a gel to see if we could see, a protein gel, to see if we could see protein cross-linking. And so what we saw is when we look at the fluorescent signal, you can see like some really faint bands at later time points. That's what this B is, a later time point. That seems to suggest that there might be some cross-linking. But if you just look at a regular Kumasi stain, it doesn't really look super compelling. And we thought that maybe this had something to do with the reactivity of our chemical probe. So Cordiana looked at a couple of analogs of this probe and found that only 2 of them seem to have any fluorescent labeling at all. And of those 2, only 1 can give us this banding pattern corresponding to high molecular weights, oligomers being captured with our chemical tool. And so there's a lot more optimization to do in terms of chemistry, and we also are optimistic that we can take this and use it to develop a high throughput screening assay so that we can screen specifically for molecules that change the conformational dynamics of proteins. I also want to briefly highlight other work that's going on by 2 other second-year students in my lab, Aaron, who is using chemical biology to also help study these oligomers so that we can learn more about protein structure function in this system, and Isha, who's looking for molecular glues that can take a specific protein of interest and target it for chaperoning by DNA JB6. And excitingly, I also was able to recruit 3 new graduate students to my lab this year who will also be working together towards these 3 goals. In the long term, we hope that this leads to better diagnostics and better therapeutics that, had they existed 30 or 40 years ago, would have prolonged the lives of the people on the first slides, people like Rosa Parks and Stephen Hawking, but more importantly, that if we have them now moving forward, that we can really improve the quality of life for people who are diagnosed with protein aggregation diseases. And with that, I'd like to thank my entire lab, I'd like to thank my funding sources, and I'd like to thank all of you for being a great audience.
We have a couple minutes for questions for Oleta.
Is it automatically on?
Okay. What was the dynamic range of CC3B? So you showed CC0, right? Did the dynamics change with CC3B?
Great question. That was brand new data. Heather will tell you that I actually sent my slides in late to try and get this on the slide. But Cordiana's working on that right now, so hopefully we will know, and hopefully it's an improvement.
So if you're thinking about this as a potential therapeutic, have you thought about when? Is it going to be prophylactic, or is it going to be early in the disease, or even late?
Yeah, great question. So I will say for the probe that I showed, we are actually thinking about it more as a diagnostic. And so the idea being that we would be able to detect things earlier than, or detect protein conformations that are prone to aggregation far before we can now, and that this could be something that would be complementary to some of the serum-based detection methods that are the gold standard in the clinic for diagnosis right now.
That was really nice data. Thanks for sharing that. I'm just curious if there was specificity to the protein that you had showed that there was binding of the probe.
Yeah, great question. So it's my favorite chaperone term is selectively promiscuous. And so, in fact, actually, DNA JB6 and one of its very close paralogs are used as tools to detect cell stress globally, even if they're not really endogenous or biologically relevant clients or substrates. But it does seem that there's some selection for proteins that tend to aggregate. So most proteins that we think about as aggregating in the diseases I showed on the slide, we see it. There's also new data about DNA JB6 being important for suppressing the aggregation of components of the nuclear pore complex as it reforms. And so you can see that as a function of aging, DNA JB6 becomes more important, but the endogenous clients of the protein are less, not well understood. And I don't know if you asked if this is what you were thinking as well, but in terms of the molecule, we're actually hoping to be able to engineer selectivity for specific proteins so that we can make it a more broadly applicable tool.
All right, thank you, Oleta.